So, Hud, are you with us?
Yes, ma'am, I'm right here.
So, please go ahead. We are listening.
Well, Ava, thanks very much for that kind introduction. Much too flattering, more than I deserve. But Vanessa, thanks so much for accommodating me in my travel schedules by allowing me to come in and do this talk by Skype. We'll see the challenges of doing this, but let's get started. So I've called this CDG Discovery, Diagnosis, and Outreach. And you can all see my first slide. And I'm going to go through several points. One is about what are our lab's goals and how do we get started in all of this? And then how do we and others, our collaborators, discover CDGs? And then how do we communicate with the families? Because I think that's a very important aspect. And how do we interact with other doctors and researchers? And I'll sort of finish up by saying about what advances and slows our progress as we come along, I think, in our goal to help you and help families, because I think that's, to us, the most important thing. And then we'll have time for questions after that. So, first of all, who are we? There are primarily two people in the lab that are sort of constant. And myself, I'm a PhD, not a medical scientist. I'm not a physician. I'm a basic glycobiologist, and I have an interest in glycosylation and glycosylation disorders for over 35 years. So this is close to my heart. And Bobby Ng, who's an extraordinary scientist. He's an insightful experimentalist. He's an exceptional technician. And I think that you would all benefit from meeting him, and maybe you can at some point. He really maintains the contact in our lab. So we share that same interest in both science and helping families. So our overall goal is to, as basic researchers, it's to discover new types of CDG, because as fundamental scientists, we have to show that we're making progress, not only being knowing those things that are already discovered, but yet moving science forward. So we try to understand the underlying science, and then also, in the course of that, make a difference for patients. So what's our entry point? How do we get started in all this? Well, most of the time, it's a DNA sequencing, which I think you've probably heard about by this point in the meeting. So DNA sequencing results or transferrin testing will be made aware of it. And where does that come from? Well, that's from families themselves who can contact us. And if not the families, then physicians will contact us. And together, we all have to work together. So, excuse me, how does this start out? Well, what's transferrin? I think you've probably, in the previous talks, already heard about this. Transferrin is a glycoprotein that circulates in the blood, in the plasma, and it's glycosylated. So it turns out to be a really good marker. And here, if you can see my cursor, Ava, say yes if you can see my cursor.
Yes, we see it.
Okay, great. All right, thank you. So this is a schematic of a molecule of transferrin. That little squiggly red line is the protein. And these big things up here in all the colors that look like an aggregate of the rainbow are the two sugar chains. And this is what it looks like on one of our super balances that we can weigh individual molecules. So this is what transferrin looks like in control subjects, in normal people. And then if there is a glycosylation deficiency, you can see that what happens is we still have the squiggly lines, but now one of those two chains of sugar chains is missing. Or in some cases, they're both missing. And this gives us a sort of a picture that there's something wrong with glycosylation. We don't know what it is, we don't know what causes it, but we know there's something wrong. And we can also find that sometimes there are differences here in this early region which would show a loss of individual sugars. So by diagnosing the kind of pattern that you get from this transferrin presentation, we can begin to figure out what sort of glycosylation disorder might be present in your kids. So I'm sure everyone is aware of how important the interaction between basic scientists and physicians is. That interaction, that basic science drives medical discoveries. And those discoveries are the things that eventually become understanding of the disease and hopefully in the future therapies. But one of the things that we unusually as basic scientists have been able to do is to involve the families. And that's what as I mentioned, sometimes families will contact us directly. So even though commonly families will be only working through their physicians, what we have now is direct contact with families. And that's critically important for us because over the years, and many of you sitting in the audience will know that we've formed ongoing relationships. Some of these things go on for 15 years or so. So I'm sorry I can't be there to see you all. So how do we communicate with families? Well, as I said, either the family or physician will contact us. But then in order to go forward, in order to be a player in this very important arena, we have to obtain what's called informed consent. That means that the patients and the family sign documents that give us permission to talk to each other. Because remember, your relationship with your physician is confidential. And so we have to be brought into that system to be allowed to access your information. So that can be clinical information. It can be things that a basic researcher and your physicians will talk about. And then we'll try to evaluate the results that come in, either from DNA sequencing that we talked about, or from transferrin. So sometimes based on this, and signing the documents and having that in an opportunity to have discussions, we can see that we may be missing certain things. So usually in our lab we'll request skin cells and sometimes DNA, which these days can be taken just from a little cheek swab. So if any of you have gone for the more popular form of genetic testing that's out there, which is 23andMe and things like this, you know that it's really quite a simple procedure. We'll also get serum from the patient and sometimes other family members. And so this helps us in the diagnosis. So how do we discover CDGs? So once we have the patient samples in hands, then we have certain assays, sort of routine assays that we can do to either rule out or in some cases rule in suspected glycosylation defects. And we'll do this from an aggregate of our understanding of past cases, clinical presentation, discussions with the physician. So then we usually suggest testing of a panel of CDG-specific genes. And this can be done at a few places in the US, but typically this is now moving away from individual panels and more toward whole exome sequencing, which essentially gets all of the DNA that's in your body that we think plays the most important roles in actually determining what happens in your body and what is one of the determiners of disease. And so at that point, excuse me, if the CDG panel shows that it's a known gene, then because we're more research-oriented, then we may stop working on it at that point, especially if it's a mutation in a gene that is already known and that mutation is already known. Then that becomes more of a diagnosis. So on the other hand, if that shows that there is no obvious defect in genes that we know, then we send a trio of DNA. That means the proband, in other words, the child in most cases, and the parents to the Center for Mendelian Genomics at the University of Washington. And they do exome analysis. Now, here's the thing, and many of you have probably experienced this, analyzing the results. First, the samples come in, they have to be quality controlled, they have to be checked, they have to get in line. So that in itself can take a long time. And then after the results are generated, it's not always so quick to find out what the likely defects might be. And then if we find a potential defect, then that can take even longer to figure out. And that's because now the sort of the basic science oftentimes has to come into play. A gene that we think maybe we knew about, we have to design assays for to figure out how this works in cells from the patient and figure out ways to show that that gene is actually defective. And as I say, that can take a long time. So how do we interact with other doctors and researchers? Well, if we find a new type of CDG, then we'll ask other physicians and other scientists if they've also found a similar sort of disorder. And that may sound simple, but sometimes it's not. And the reality is that those scientists who are involved in research are in an academic setting. And the scorecard, if you will, is being the first to discover something. So it's prestigious, it's treasured, it can advance your career, it can get you grants. It's benefit to the individual. Second place, that's much less value. Now, on the other hand, you as parents don't necessarily care about our little issues as scientists. You want something for your kids. You want to find out what's going on, and you want scientists to cooperate. And I firmly believe that the families are a force, an empowered force that can catalyze the change of getting more physicians and more scientists to cooperate with each other. You're the big stakeholders. And you can do this by stressing the need for collaboration. And if you do this, I think it can change the world. There's already a lot of changes going on in research. You're an essential component of that. As evidence of the changing times, a guy named Eric Topol, who actually is right across the street from me at Scripps, wrote a book that just came out in January of this year. It's called The Patient Will See You Now, not the doctor will see you now. And what's important about that? It says that the face of medicine is changing. Instead of the doctor being the all-powerful person who knows all and may share that information with the patients, or in some cases decides not to, that's gone. Thousands of years of the paternalistic medicine is changing. And why is that? Because there is availability of information on the web. Families find their way to my lab and other labs because they can go on the web. They can be proactive and find out what's going on. And in fact, they do that. And the other thing is there are wearable devices now that can actually monitor many of the things that you would have to go to a doctor's office for. So one of the examples that Eric gives in the first chapter in his book, I'll come back to here in a few minutes, but it involved a disorder that we've been involved with too, and you'll hear more about this in a few minutes. So you're empowered. So what kind of thing, what advances can slow our progress or advance it? So from my perspective, if we become involved with the family, we really depend on you for efficient communication with us, with the physicians, and then every physician we need to have quick response from. I can tell you that there's been some instances where that doesn't happen. Other ones are just exemplary, and we move forward fast. And we think that every family should receive and or ask for a periodic update. If we're going to operate with your kids, we got to give you something back. We owe you that. So every family should receive or ask for periodic updates. Now, where's the balance where you're maybe asking too often? I know that sometimes that we'll have people who call, and believe me, we return all of the calls and emails. Bobby especially is exceptional in this. Now, So we like that interaction and ongoing communication. And sometimes, unfortunately, suspected defects turn out really not to be the defect. And you say, well, how can this be? I thought this was all established. Well, it isn't, because there are thousands of genetic changes that happen, but there's only a few of them that actually cause a disorder. And we oftentimes have to sift through those things. And being able to do this requires that period that I was talking about, where we actually have to validate that a mutation is actually damaging and causing the kinds of glycosylation abnormalities that we see. And that's what can be slow. And we work at it. And every lab that is involved in this keeps working at it. But as a research lab, we're not a diagnostic service. So we can't sort of respond in the way that a sort of fee-for-service laboratory or your perhaps other contacts can respond. We don't receive any money for this. Our job is research. So any results that we produce, the clinical labs have to be able to replicate those results and then sort of put the stamp of approval on them. We're research. So what do we usually do? Well, if we get DNA from patients with abnormal transferrin, glycosylation, showing abnormal glycosylation, well, what we can do at that point is to send the gene sequence to the University of Washington, or I should say send genes for analysis to University of Washington Center for Mendelian Genomics, and then we go through those results, and Bobby's exceptionally good at that, and we sequence the biochemical information that we can extract from that. In the last couple of years, we and our collaborators together have actually found 7 new CDG defects. So I think we're doing pretty well. Bobby, for instance, in our lab has compiled these statistics that, in fact, we've headed toward almost 300 fibroblast lines from patients. And we found defects by analyzing these in over 200. We've done this thing called whole exome sequence with the University of Washington in 64 cases, and we've solved a little better than half of them, but we still have unsolved cases. We've done all right in discovering new disorders, but we've also got potential new disorders in the pipeline. And this is the thing as basic scientists that is of great interest to us. If you look at what's happened in the CDG field for over the last, let's say, 20 years, you can see that there's been a tremendous growth. And this has especially occurred in this orange set of blocks, which are the N-linked glycosylation disorders, the one that transferrin mostly picks up. In fact, in 2013, there was a new disorder published in the literature on average every 17 days. So this is far outpacing, I think, any other specific kind of disorder that's known to be caused by genetic defects. So we're at the very front in many ways. But transferrin doesn't always detect all kinds of CDGs, and in fact, some CDGs that actually alter what we call glycosylation homeostasis. In other words, the balance and the intersection of how different glycans are made, how they're used by the cell, how they're used by the body. This is an evolving issue, and that's what science ought to do. Science ought to contribute more knowledge as we go along and link things together that we hadn't seen. And sometimes these disorders will occur in several pathways, and they don't change transferrin. And even as we found in some recent work, some of the predicted changes in transferrin, those don't occur either. Now, we know that CDGs, as I say, can disrupt homeostasis, and that just simply means the balance within a pathway. And I'm going to finish up by a specific example, which you may have already heard about, called NGLY1. And this is a disorder that we became involved with, and I think you've probably already heard from Matt Might, but let me perhaps put a little bit of a different perspective on this. This was when the Might family contacted us, we became aware of this blog. And I don't know if Matt put this in there, hunting down my son's killer. Found my son's killer. It took 3 years, but we did it. This was a blog that got over 2 million hits. Now, Matt knows his way around the internet, knows how to blog, and he's one hell of a writer. This guy is compelling. So we found, or I should say the Goldstein lab found, a deficiency in something called NGLY1. And this is a disorder of deglycosylation. Now, it's related to glycosylation, and you probably heard Lynn Wolfe already talk about this. But because there were this many hits, it attracted the attention of some prominent science writers. And on the left, you can see that this was an article published just a little bit over a year ago by an author named Seth Mnookin. Published this in the New Yorker. It was called One of a Kind. And this is Bertrand, who you've probably seen at the meeting and heard about. And then it was also a similar article was published in Der Spiegel, which is an equally prestigious magazine in Europe. And of course, here's the family that you've probably now seen at the meeting. So what's important about this? Well, it shows what can happen if the family goes out, not just finding a single patient, their own, but what Matt and Christina have done is to go out basically worldwide and say, Bertrand is not one of a kind. Now we know of about 35 patients, and some of them, I believe, are there at the meeting. This, in a very, very short time, is a phenomenal operation. And it attracted the attention at high levels. And in fact, there was the announcement of the Precision Medicine Initiative in Washington several months back. And Matt, because of this placement and because of this notoriety, was able to meet this man here, who is Francis Collins, and he, you'll notice the little squiggly DNA thing there, is the director of the National Institutes of Health, which funds almost all the medical research, probably half of the medical research in the United States, certainly the basic research. So we got to meet somebody who, you might say, gives us a well-placed advocate. And now this can increase awareness for CDG. But Matt didn't stop there, because he got to meet another sort of well-placed advocate, you might say, at the White House, and now CDG is known at the very highest levels in the United States. CDG, President of the United States, we got a connection. That's important. That makes a difference for us. What's important? I think advocacy and education, and that means you as the major stakeholders, as the parents, you need to advocate and educate physicians, scientists, other families, and decision makers. We've already got one, but we're not done yet. We got to keep going. We have to have inclusive alliances of all the stakeholders. And this means cooperation among physicians and scientists and families. And we're going to bring in those decision makers, those people who have the ability to make a difference for us and you and the families. And one of the things that you can do as families is to support fundamental discovery research, because that's what's going to create the hope for the future. And if there are treatments to come, and we sure hope that there are, this is where we're going to find it, in fundamental research. So I'm going to stop now, and I'll take your questions.